I'm Francesco Strozzi. I will talk today about how we applied AlphaFold and deployed it at scale to try and decode the human gut microbiome proteome. So first of all, bear with me. This is probably the only non-bright white slide in my talk. So yeah, I know. If you need to put on sunglasses, but do it, it's okay. So a couple of words about Enterome, the biotech company where I work. So what we do in Enterome is basically try to develop a new class of biotherapeutics compounds. Where we hunt these new biotherapeutics is from the human gut microbiome. Specifically, we hunt for short peptides and small proteins. And we have different lines now in different programs running now. One is called oncomimics, another endomimics, allergomimics. Basically, we try to look for small peptides and proteins that are produced by the bacteria in the human gut microbiome that can mimic or trigger some immune targeted response in the human body, specifically against, in the case, for example, of oncomimics, some type of cancer. So things are going quite good for these kind of programs. We have four actually programs which are in phase 2, 2 clinical trials that should end phase 2 next year. So as we speak, there are patients that's been treated with these compounds in immuno-oncology spaces, and the results in the clinic look quite promising. So if you're interested, go and look at the website. We have much more information about this than in my presentation. So how we do look for these kind of molecules and peptides and proteins? Well, upstream of the more traditional drug discovery programs the company is running, there is the computational part. So how we do that is that we compiled during the years a huge microbiome database, so a large collection of more than 20 million proteins that are encoded by the bacteria in the human gut microbiome, and then through the combination of different tools, bioinformatic tools, we hunt for genes and proteins, and then we try to annotate and infer as much information as possible about what these small peptides proteins do, and if they can be a good candidate for our programs and to target specific disease. As I said, specifically immuno-oncology and GI tract disease. So this is where AlphaFold arrived. So last year it was published this big paper. The team behind, so DeepMind, released this deep learning method that tried to solve a long-standing problem, which is basically, try to go from the protein sequence to the structure, trying to predict the 3D structure of a protein just using the sequence. It is and it was a very, very important breakthrough. What basically the team did was to try and develop and make a very complex deep learning method to learn the links between conserved evolutionary blocks in protein sequence and link those to 3D conformations. So at the very beginning, when the pipeline was released, it was released like a monolithic pipeline. So there is this initial step that is needed to identify these evolutionary conserved blocks that require you to run a multiple sequence alignment with your query protein against a huge database of known protein sequences. And then there is the more GPU-based deep learning part. So the most obvious thing, the initial thing we needed to do in order to develop a pipeline that was scalable, because we wanted to apply these to our large collection of millions of protein sequences, was to decouple these two parts. Most of the work here is inspired by the ColabFold, which is a paper and a web resource that was published a few months ago, where basically Martin Steininger team applied the MMSEC2 algorithm for fast multiple sequence alignment of a query protein sequence against a huge index. As you can see here, the index is quite huge. It's a 1 terabyte index of hundreds of millions of proteins coming from different resources, UniProt, PGD, Magnify, etc. So the pipeline is basically composed of few key steps. The input sequence is your protein sequences. Then there is the index that need to be loaded. The MMSeq search that need to be run to generate multiple sequence alignment. And then this part, specifically the multiple sequence alignment search and generation, is a very, very CPU intensive part. So this part can and should scale on thousands of computing cores if you want to, of course, run a large batch of search against many thousands of proteins. Then there is the second block, which is basically the very proper AlphaFold part, which is basically taking all the multiple sequence alignment, extract the features, and transform the features in a way that all these conserved blocks can be understandable by the deep learning method they developed, and then run the actual features through the deep learning network and generate a prediction. So in the end, we get a PDB file with the 3D coordinates of the protein in 3D space, of course, and with the probability score. This second part is very GPU intensive. So as I was saying, I mean, the AlphaFold pipeline was released as a monolithic block. In order to make this runnable at scale, it is absolutely needed and necessary to decouple these two steps. One is very CPU intensive and the other is very GPU intensive. So where we are now, so at the moment, we processed more than 400,000 proteins. This number is just not because we couldn't process more. We are on the cloud, so yes, there is a budget limit, of course. But this is the fraction out of our huge database we work with that is the most interesting for us to start to look at and try to hunt for this new next candidate for our drug discovery pipelines, because these 400,000 proteins have the characteristics already based on the sequence in itself that are most interesting to us. In terms of prediction, so here is just a quick, simple histogram showing you the metric, the probability metric that is the output of the 3D structure prediction from AlphaFold, which is called a local distance different test. In the output from AlphaFold, this becomes the probability local distance difference test, or PLDDT, which is a very straightforward acronym to pronounce, especially if you are not a native English speaker. And so we just call it the score. This score gives you basically the confidence that was produced by the deep learning method in assessing the 3D structure, and is a superposition-free score that was developed years before AlphaFold. So as you can see here, the average score for this batch of proteins we started to predict out of the human gut microbiome, it shows that, I mean, anything above 60, 70 in this score is considered to be starting to be quite a good prediction in terms of accuracy. So even if there is still a fraction of proteins that get poorly predicted, the overall majority of the proteins we pass through the model get quite an accurate prediction overall, so which gives us a good starting point to start analyzing all this structure. So how we implemented this, so of course with Nextflow, we'll be surprised if it was otherwise. So this architecture, I believe that when we started discussing batch and Nextflow a few years ago, everything was so new. Now I think that for many people here in the audience, this is like looking more and more familiar, especially if you are running your pipelines in the cloud, and especially on the AWS cloud. So as already discussed here, we have Nextflow orchestrating the whole thing. We have built containers to include all the tools that we needed, AlphaFold and MM6. We deploy this through batch, and we store our data, input and output data on S3, but more especially using the FSX Lastre file system for the index. I will come back to this in a few seconds. We rely exclusively on spot instances, and you should too. I believe that if you work in R&D and you are even a larger scale organization, there is no point in not using spot instances unless you have like very, very specific needs of having like 10,000 cores up and running 24/7 every time, which is rarely the case in research and discovery. So a few details and information about the technical challenges and the execution times of this pipeline. So as you can imagine, considering that the very starting point and quite an important part of the AlphaFold pipeline is actually not running AlphaFold itself, but generating the multiple sequence alignment at first, managing and loading and charging the index, which is a 1 terabyte, actually there are 2 index, but the indexes are 1 terabyte in size, is quite a bottleneck. So we spent a few months and tried to run different tests and benchmarks on the AWS cloud to try and identify the best resources, both in terms of price and performance, and the most useful resources we could use is the FS6 on Lustre. So this gave us high performance to expose and provide the index to thousands of instances at a reasonable cost, and the performance is really, really good considering the size of the indexes. In terms of the execution time, so once we have dealt with loading and presenting this big index to the computational instances, the multiple sequence alignment really is blazingly fast. Of course, this MMSeq behind, if you work with this tool, you know how incredible it is. So it just takes a few seconds per sequence to generate a multiple sequence alignment, and then running this MSA through the AlphaFold to get the actual 3D structure prediction takes few minutes per sequence, depending mainly on the depth of the multiple sequence alignment and on the length of the sequence you want to predict. The hardware we used could be interesting to people trying to do the same thing or that are doing the same thing. We rely on Graviton instances, were just mentioned by Matt in the previous talk, to run the multiple sequence alignment, specifically the RC6G, because they provide a lot of memory, so we can partially charge the index in memory as well, once it is loaded from the FS6. And to run AlphaFold itself, we use the new G5 instances, which are incredibly fast compared to the previous generations. When we started this project, end of 2021, we relied on the G3 instances, or G4. G5 is really a leap forward in terms of performances, and as you can see, we on average just require a few minutes per sequence to run the prediction. So just to conclude, few thoughts about building this pipeline and running this pipeline at scale in the cloud. So AlphaFold is very computational intensive, period, because you need to combine a very CPU intensive part, the multiple sequence alignment, the index, and then a GPU very specific part. So running this on premises requires you to have, of course, very specialized hardware, GPU, a lot of GPU servers and machines, in order to can keep up with the index size. Otherwise, you can run it through the cloud. It's probably easier. And in terms of cost, I don't know, maybe it's a question that could come up to run these. Probably was 200,000 sequences. We spent like 4,000 euros with everything considered. So it's not that bad, actually. Even if, of course, there is a lot of effort now from the big databases, EBI, UniProt especially, have released 200 million predictions for the proteins that are known and present in UniProt. We still needed to have this pipeline in-house because we need to process all the protein sequences that are not part of the public databases, which it's a lot of proteins that are not in the public databases, especially in the human gut microbiome. So we needed to have these in-house to process our own data. For us, it's a very important new brick in our computational drug discovery pipeline because it allows us to explore a complete new dimension in terms of similarity search, so moving from annotating sequences and functional domains to try and compare structure of proteins and look for similarities between human gut proteins and human proteins or ligands or receptors, for example. Even if AlphaFold, this is just my take on this, even if AlphaFold, it's an incredible, huge step forward in this space, and it's very useful to have the protein structure for unknown proteins. Just having this information will not allow to directly know the function of the protein, which is what we are looking for. So there is still a lot of work required, and probably you could not skip the wet lab part screening these pipeline to see what they actually do, even if you know the 3D structure, which is anyway a very, very important and useful information to have. So that's it. Thank you very much. I would like to thank also all the people from my team that worked and contributed to this and do an amazing work day after day. Thank you very much.
Great. Thanks so much, Francesco. It's great to see some multiple sequence alignment here as well. You have a bit of background is that obviously Nextflow was made with Cedric's lab, and we did a lot of multiple sequence alignment, of course. Question from Saba, have you considered other de novo protein structure prediction methods like OmegaFold?
No, not yet. So we just wanted to start with, make this a reality, so being able to run AlphaFold first, and then I had discussions with the people from Cedric's lab about the NF core protein fold initiative. So maybe the idea is to have a centralized pipeline, Nextflow pipeline, to include not just AlphaFold, but other tools in the space. That could be a very, very good idea.
Awesome. And Rob is asking, what were the sequence features that you were looking for to help prioritize those first 400,000 candidates?
We can have a discussion later, maybe not
when
I'm recorded.
And then just like, so something I was wondering is what do you actually do with those predicted structures then? So you make the prediction and then you've got a lot of them.
Yes.
How do you kind of go, what's the kind of next step from there?
The next step is structural alignments. So basically there is the new tool, again from the Steinhager group, FoldSeq, which allows to compare 3D structure at scale very fast, very accurate, to perform structural alignments and see for and look for local or global structural similarity. So this is the first step for us then. And then try to link all this information with all the rest of information we have already on the protein. And if we screen these proteins in the lab, then also include that information.
And ultimately get some kind of function.
Yes,
functional
exactly,
assignment.
Exactly. Or a possible prioritization of these proteins based on all this information combined together. Yeah.
Cool. Awesome. Thanks a lot, Francesco.
Thank you.